Happy learning.
Hi, welcome to our Pharma Topics channel. Welcome to the Pharmacological Screening Methods Unit 2. Before entering into the research in pharmacological screening, let us know some basics which is given in this presentation. First is selection of species for research. Species selection is based on the behavioral or biological characteristics related to the study of interest. The selection should yield accurate and interpretable results. Species selection is based on literature review. Species selection is also based on requirement of least number of animals to complete the study as per the CPCSCA guidelines. Selected species should have least chances of biological hazards. There should be some appropriate analogy. That is, the investigator must ensure that the species or tissue selected should have a similar functioning as that of the target species. The transfer of information, that is, the data from one species, should be easily interconvertible to the another species in a simple or modest way. Generalizing the results. The animal selection should be able to generalize the results to the other species with minimal variations, such as cultural, environmental, and dietary variations. Ethical implications. It is generally recommended by CPCSCA that if alternate methods are available, they should go for alternate methods, such as cell lines, microbial studies, or computer simulations. Cost and availability. Costing not only involves purchasing or the breeding cost, but also includes the hidden charges such as maintenance of the animal house, that is animal husbandry, and other experimental charges. Housing. Example, mice is more feasible species when compared to the species like rabbits or guinea pigs, because they can be housed in a larger number of cages and needs minimal animal care and husbandry. Stress factor. It is one of the important factors which can affect the outcome of results. Stress can affect the physiology, biochemistry, and behavior of any animal. Stress can occur during transportation, overcrowding, handling, and manipulation. So, the animal species that are more stable against the stress are more feasible for the study. Genetic aspects. Continuous inbreeding can lead to genetic drift, which can affect the outcome of results. Biological properties. Example, a rat would be best choice for biliary studies due to absence of gallbladder. It does not have a vomiting center, so antiemetics could not be screened with rats. Adaptability of the species. The guinea pigs have highly inaccessible blood vessels, so they cannot be used for studies where more blood sampling is required. So they may go for rabbits or rats. Size of the animal. The size of the animal not only impacts housing and husbandry, but also it is very important in consideration for tissue sampling and blood collection. The next is lifespan of the animal. Studies with a lesser time are more feasible with animals that have a shorter lifespan, like a rat or mice, which is approximately 2 to 3 years. Whereas for longer duration studies, animals like dogs or primates could be chosen, which have a lifetime of roughly 12 to 30 years. Gender. Studies which are performed in both genders have applicability in male and female in humans, whereas studies conducted in female animal species exclusively, such as teratogenicity, contraception, lactation, and breast cancer, should be conducted in the same female species. Progeny. Some studies are designed on the progeny. So the number of progenies produced by an animal is also considered in choosing the animal species for research. That is the number of next generations. Next is vehicles. The most commonly used vehicles are distilled water, normal saline, 50% polyethylene glycol, 0.25% to 1% methyl cellulose or carboxymethyl cellulose. Volume for oral route, it should not exceed 1 ml per gram or 10 ml per kg body weight of the animals. Preparation of the stock solutions. It is calculated by multiplying on both sides by a constant volume. Whatever volume is needed and whatever dose has to be administered, it has to be multiplied on the both the sides. For example, if we need 20 mg in 1 ml, multiply 40 on the both the sides. So 800 mg in 40 ml can give the required concentration. The next is objectives of research. To promote the human care of animals in biomedical and behavioral research. To perform testing that will enhance well-being quality in pursuit of advancement of research. Quarantine, it is a separation of newly received animals from those already in the facility, that is the animal facility, until the health and possibly the microbial status of the newly received animals have been determined. An effective quarantine minimizes the chances of introduction of pathogens into established colony. Duration for small animals, that is for mice, rats and rabbits, it is 1 week. And quarantine for large animals, it is approximately 6 weeks, cat, dog and monkey. Quarantine helps in limiting exposure to the zoonotic infections. Quarantine also prevents interspecies disease transmission and eliminates anxiety and possible physiological and behavioral changes due to interspecies conflict. Restraint devices cannot be used simply for convenience in handling or managing animals. The period of restraint should be minimum to accomplish the research objectives. Animals to be placed in restraint devices should be given training to adapt to the equipment. 3 R's in animal research. Replacement. If possible, replace the methods in animals with in vitro or in silico methods. The next R is refinement. To minimize the potential pain, suffering or distress to the animal used during the experiments. Then reduction. If not possible to select any other in vitro procedure, then minimize the number of animals used for experimentation. Actually, there is a 4th R, that is reuse. It is applicable for large animals after experimentation. For example, dogs could be adopted by those who want pet animals. So they could be adopted after that is experimentation. Treatment groups in the animal research. There should be a control group or normal group. This group is used to assess the impact of external variables, that is environmental or other possible unwanted factors other than the tested drug on the biological response. Then vehicle control group. This group assesses whether the vehicle alone produces significant biological effect or not. Sham control group. It is employed to assess any significant effect of surgical procedure or any other special treatment such as intracerebroventricular injection in animals. The next is positive control group. These are the groups in which a standard drug with a known biological effect is administered. In this group, biological effect is compulsorily expected. Comparative groups. In this, a direct comparison is made between the biological effect of the tested drug and that of the standard drug. Effective first line or a drug of choice for that disease. Validity and reliability in animal research. Validity, it refers to how well a test measures what is supposed to be measured. Face validity, it refers to similarity of symptoms produced in the disease model and naturally occurring disease in humans. It means how closely an animal model resembles a human disease. Next is reliability. It is the degree to which the assessment tool produces consistent and reproducible results. Crossover design in animal research. In crossover design, the same animal is assigned to receive 2 or more treatments in a particular sequence. In this group of animals, they receive the drug treatment in the first period. Thereafter, suitable long washout period, the same group receives standard drug treatment and so on. The greatest advantage of this design is that the individual groups acts as their own controls. Another advantage is lesser number of animals could be used for the research. Disadvantage of crossover design is difficult to interpret the results. Pre-anesthetic medication before anesthesia in animal research. Before the actual anesthetics for surgery, the animals are prepared for anesthesia by overnight fasting and using pre-anesthetic medication. This reduces the parasympathetic stimulation of cardiopulmonary system and reduces salivary secretion. Atropine is the most commonly used anticholinergic agent. CPCACA guidelines for surgery in laboratory animal research. Multiple surgical procedures on a single animal for any testing or experiment are not to be practiced unless specified in the protocol that is approved by IAEC. Maximum experimentation duration in laboratory animal research. Animals should not be subjected to experimentation for more than 3 years as per CPCSCA guidelines. Ethical issues associated with the blood collection in the animal research. It is a controversial method because it produces a discomfort to animals. Inflammation reactions are produced at the site of puncture. It takes approximately 4 weeks to heal from the damage created. There is also possibility of formation of hematoma in the retroorbital site. Nowadays, retroorbital blood collection is not applicable as per CPCACA guidelines. The pressure of hematoma may compress the optic nerve to produce blindness. Biostatistics in laboratory animal research. Statistics, it is an important tool in pharmacological research that summarizes experimental data to enable us to conduct hypothesis testing and to make conclusions based on the results. Variable. Variable is any characteristic, the number or quantity that can be measured or counted. Discrete variable. Example, the number of animals that die when 18 animals have been tested for LD50. This is a discrete variable. Continuous variable. Example, weight of an animal can have value between 190 and 210 because in between, then it is a continuous variable. Then ordinal variable, that is ranking the effect of test drug as low, medium or high. It is an ordinal variable. Next is nominal variable. For example, gender cannot be ranked. That is a nominal variable. Then variance. It is the measure of how far each variable value in a given data differs from the mean value is called a variance. Standard deviation, it is another way of representing the variance, which is square root of variance. Standard error of mean, it is a type of standard deviation which describes how accurate is the sample mean with respect to the population mean. Accuracy, it is the representation that how far the experimental results agree with the true or the actual value. Precision, it is the measure of reproducibility of an experiment. Bias, it refers to the systematic variation of observed values from the true value. Symmetric distribution, that is the distribution of data that has same shape on both sides of the center, is called symmetric, which is also called binomial distribution. Hypothesis, it is an assumption or prediction or tentative answer of a research question or questions. Placebo, it is a sham substance or treatment that does not have therapeutic value. Research question, does the new drug alter the blood sugar levels more than the placebo? Is a research question. Null hypothesis, the average effect of the tested drug on the blood sugar level is equivalent to that of average effect of the placebo. There is no effect, then that is a null hypothesis. Alternate hypothesis, the average effect of the tested drug on the blood sugar level is not equivalent to that of average effect of placebo, then it is a alternate hypothesis. P-value, it is the probability of rejecting or accepting the null hypothesis of the research question. It determines the significance or non-significance of results. P-value less than 0.05 indicates a strong evidence against null hypothesis. So the null hypothesis is rejected, which means the test drug is different from placebo. P-value greater than 0.05 indicates a weak evidence against null hypothesis. So the null hypothesis is not rejected, which means the test drug is not different from the placebo. Difference between p-value and the confidence interval. P-value indicates a statistical significance. whereas confidence interval represents a clinical significance. Degree of freedom, it is the difference between sample size and number of parameters estimated. For example, sample size is 6, then the degree of freedom is 6 minus 1 equal to 5. Type 1 error, that is false positive or alpha error. Null hypothesis is actually true, but rejected as false by testing is alpha error. Type 1 error, false negative, that is beta error. Null hypothesis is actually false, but it is accepted as true by testing. This is type 2 error. It is more dangerous than type 1 error. There are two categories of tests which are applied in statistics. One is parametric tests. It is applicable for numerical data which is normally distributed, that is students t-test, one-way ANOVA, repeated measures ANOVA, that is two-way ANOVA, three-way ANOVA, or MANOVA. Then Pearson correlation coefficient. Then non-parametric tests, they are applicable for non-numerical data which is not normally distributed, that is chi-square test, Fischer's exact test, Mann-Whitney U-test, Wilcoxon signed rank test, Friedman's test, and Spearman rank order test. The post hoc test, nowadays the softwares, they analyze one-way ANOVA followed by t-test. That means comparison between more than two groups is called ANOVA. Then inter-individual comparison, it is called post hoc test. So it is a test which is applied after applying ANOVA. Types of post hoc tests are Dunnett's test, Bonferroni test, Scheffe's test, Fischer's LSD, Tukey's range test. And the softwares used in biostatistics are SPSS, statistical package for social statistics, sigma plot, then graphpad prism, instat, biostat, and statistica. So it's a very short information of the basics of the research which has to be known to a researcher before studying or applying the pharmacological research methods, that is applying the research. So I hope it will be useful for all the researchers and PG students and the UG students who wish to carry their research in pharmacological drug discovery. I hope it will be useful for you. Thank you for listening. Happy learning. Share this to more of your friends. If you like this video, kindly press the like button. Go through all the series of videos which is available in Pharma Topics channel. Thank you.